Okay. This is James.
James.
Dane.
Dylan.
And we're tackling lesson number 3, group problems. So the first question, part A, asks us human disease, Angelman syndrome, and it's asking us if recessive A is the Angelman syndrome allele, where dominant A is non-disease allele, and we cross heterozygous male with heterozygous female, what genotypes and phenotypes of the progeny will we get? So this is what we have. We have 25% for homozygous dominant A, and 50% for heterozygous, 25% for homozygous recessive. So that the ratio will be 1:1. Phenotype will be the same, normal, 25%, 50% carrier, 25% diseased. For B, it's saying the cross between heterozygous male and homozygous recessive female, what would it produce? And for the genotype, we got heterozygous 50% and homozygous recessive 50%. So the ratio will be 1:1. Carrier, again, 1:1, 50% to 50%. For C, suppose a woman has triplo X syndrome, 3 chromosomes, but 2 of them do not have any XICs. How many bar bodies are there in her cells? So since XICs are inactivation centers, we need an XIC on each chromosome in order to activate 1 or 2 of them. So in this case, since only one has one XIC, the result will be there will be no bar body in her cell. For D, suppose you isolate a cell from a female cat with 2 X chromosomes, but
Didn't we say it was 2 for C?
No, it's D for C. I'm sorry.
Oh, no. Okay, got you.
Okay, sorry. So this cat has 2 X chromosomes, and the T6 genes on each X chromosomes are non-functional, while the cis genes are functional. So in this case, the inactivation, so the cis inactivates both chromosomes, so which will give us 2 bar bodies.
Okay.
All right, so for question 2, it's just simple fill in the blanks. A is SRY is an example of a Holland-Drake gene. B is MYC2 is an example of a gene found on both the X and Y chromosomes. C, a cross between this right here, a male mouse with that genotype, and a female with this genotype. And we came up with the phenotypic ratio of 1:1, but the genotype has a ratio of 1:2:1. So for D, in the sex-influenced pattern of inheritance, an allele can be dominant in one sex, while the same allele is recessive in the other sex. And we have the ram example for that one with the horns. E, genomic imprinting is an epigenetic process that occurs during gamete formation, while dosage concentration is an epigenetic process that occurs during embryogenesis. For F, one of the topics discussed in lesson 2 involved the formation of a double-stranded RNA molecule. What 2 molecules form 2 halves of this double-stranded RNA? Answer is the XIST and the TSIX. For G, when reciprocal crosses give identical results, the alleles that control the trait in question are located on autosomal genes. When reciprocal crosses give different results, the alleles that control the trait in question are located on the X chromosome.
Okay. For number 3, we are talking about OTD, and it's an X-linked recessive disorder producing mental deterioration. So the first question is, what is the genotype of the woman's father? The woman's father has it, and the woman's mother is homozygous and is unaffected. So A, the father's genotype would be a recessive R on the X, and then Y, and the mother would be homozygous dominant with the normal genes. normal allele, sorry. If the woman has children with a man who does not have ODT, what is the chance that their first child will be a son with ODT? So I did a Punnett square here, and so 50% would be sons, and only one of those two would have the OTD, so it's a 25% chance of a son with OTD. For C, it says identify the genotype of a male with whom the woman could produce a daughter with ODT. So the male would have to have ODT because it's recessive, so a daughter would need 2 defective X chromosomes. So 1 X defected from the mom and 1 X from the dad. For D, it says consider the cross described in part C. What are expected genotypes and phenotypes of the offspring? So there's the Punnett square. The small r is defected and the large R is normal. So there'd be 25% chance of a son with OTD and 25 without OTD, and then a 25% chance of a daughter with OTD and 25% chance of a daughter without it, but she would be a carrier for that.
Okay,
Sorry.
so no problem. So we're doing question 4. The presence of beard in some species goat is determined by autosomal gene that is dominant in males and recessive in females. So question A asks, suppose that a cross produces 3 beard to 1 no beard phenotype ratio among the male offspring. What are the genotypes and phenotypes of the parents? So the parents, the father will have to, and the mother will have to be heterozygous, I'm sorry. As you can see, once they cross, these are the 3 affected progenies with beard, and this is the one with no beard. So suppose that a cross produces 1 beard to 3 no beard phenotype ratio among the female offspring. What are the genotype and phenotypes of the parents? So the genotypes and phenotypes will be the same as above. In this case, only one of them is affected, so only one will have beard and the other 3 will not. So for C, suppose a cross between a male with a beard and a female without a beard produces a 1 beard to 1 beard, no beard phenotype ratio among the female offspring. What are the genotypes of the parents? So in that case, the father will be homozygous dominant, and whereas the mother will be heterozygous, and this will be the cross that they produce, so it would be 50-50. For D, suppose a male and female, both which lack beards, produces 1 beard, 1 no beard ratio among the male offspring. What are the genotypes of the parents? So for the father, we'll have homozygous recessive, for the mother, heterozygous, and that will produce this ratio.
All right, so question 5 starts off with the cats. It says, in cats, orange coat color is determined by the B allele, little b, and black coat color is determined by the big B allele. The heterozygous condition produces a phenotype known as tortoiseshell. The big B and little b alleles are X-linked. Suppose that you cross an orange male cat to a tortoiseshell female cat. What are the genotypes and phenotypes of the progeny? So you have the homozygous recessive, which is for the male, who's orange, and the heterozygote female, which gets the tortoiseshell. And so offspring, you'll have 1 tortoiseshell girl, 1 black boy, 1 orange girl, and 1 orange boy. So it's a 1 to 1 to 1 to 1. And for B, this question concerns the trait described in part A. Male tortoiseshell cats are very rare. What is their genotype? So they just have an extra X chromosome, which allows them to be tortoiseshell, so it's XXY. In fruit flies, a mutation process produces the scalloped phenotype, which is irregular margins on the wings. Assume the scalloped phenotype is the result of an X-linked recessive mutation. Indicate the genotype and phenotype of the progeny produced when reciprocal crosses are done. So it's really simple. It's just a 50-50 ratio. And for D, this question concerns the same flies in part C, but it's no longer X-linked. It's now on the autosomal chromosome, which leads to 3 being unaffected and 1 being affected.
So I thought because it was autosomal, you're gonna have a homozygous recessive and a homozygous dominant. So that
hit pause on that.
How do you pause?
You could just you might just have to stop it.